My name is Riley Weisup, and this is my 9th grade biology project on how the virus HIV reproduces and how anti-HIV medications work. First, I'm going to explain how the HIV virus is structured. The virus has proteins around it called GP120s. I'll explain more on these later. Inside, the HIV holds RNA. I forgot to add this in the first video, so here's a drawing of what holds the RNA. It is called a capsid, and it is basically the nucleus of HIV. But that's all you need to know about the structure of HIV for this video. So on to HIV reproduction. The first step of HIV reproduction is binding. HIV attacks the T lymphocyte cell, a cell used in your immune system. To start the process, it binds with the cell membrane using its GP120 proteins to attach to the CD4 and CCR5 receptors on the cell membrane of the host cell. This is a little picture of the proteins. Second step, fusion. Once the HIV has bonded with the host cell, it will then deposit its RNA into the host cell. Third step, reverse transcription. I didn't write this in, but it's reverse transcription, not just reverse. Why it's called reverse transcription is because it's exactly what it says. Instead of the normal transcription that turns DNA into RNA, this turns RNA into DNA. Now that the RNA is in the host cell, it needs to be changed into DNA so that it can enter the nucleus. The viral enzyme, reverse transcriptase, will change the RNA into DNA. Ta-da! DNA! Woo! Fourth step, integration. In this step, the HIV DNA is going to enter the host cell's nucleus. A viral enzyme called integrase will take the bad DNA, the HIV's DNA, and insert it into the host cell's DNA. It hides it so that HIV DNA will be copied with the host cell's DNA. It's hiding it here in this drawing picture. The red is the bad DNA and the purple is the good DNA. Step 5. Transcription and translation. I'm going to go through this pretty quickly because it's the same process as a normal healthy cell would go through. So here, the DNA has opened up to allow the mRNA to transcribe its DNA. The mRNA is using the blue here instead of the orange to show the uracil that mRNA uses instead of the thymine that DNA uses. The mRNA is then going to move on to the translation stage, where the ribosome will send out tRNAs to uncode the bases to make amino acids. Once the mRNA chain tells the tRNA to stop making amino acids, the amino acids will then leave the ribosome and form proteins. Shown here, they're just little squiggly things, but yeah. The sixth step, assembly. In this step, the proteins are then going to come together to form particles and then eventually a new HIV virus. They go towards the cell membrane, which is why I showed a little airplane. They don't really go in an airplane, but they travel towards the cell membrane, and that's where this step happens. Seventh step, budding. This is the final stage where the HIV virus is going to push out of the host cell, stealing some of the host cell's cell membrane, and then going to do the same process that it just did to this host cell to another harmless host cell. This whole process is sometimes known as hijacking your body because it uses the cells and their ribosomes and their energy and their amino acids to make them code for something else, which is coding for a new HIV virus and using them to just make bad things instead of good things. So to stop this, we have anti-HIV medications. These medications only work when the patient has HIV, not AIDS, which is a follow-up to HIV that actually affects your body instead of just using your body to reproduce. In this video, I'm going to be talking about two main medications, NRTIs and PIs. These medications tend to work hand in hand to prevent HIV viruses from producing antibodies against the medications. Let's start with NRTIs. This medication affects the reverse transcription step. It stops the viral enzymes from taking the HIV RNA and changing it into HIV DNA. This stops more HIV viruses from being made because the RNA cannot enter the host cell's nucleus, and the rest of the steps cannot be completed if the HIV RNA, or supposed to be HIV DNA, doesn't enter the host cell's nucleus. For the second medication, PIs. PIs affect the assembly stage. They stop the HIV proteins from forming into HIV particles and then into an HIV virus. Both of these work together to stop HIV from reproducing and affecting more cells. Doctors will tend to prescribe both of these medications to you at the same time, telling you specific times and amounts to take them so that the HIV virus does not create antibodies against the medications. Patients who have HIV are most likely going to be taking these medications for the rest of their life to ensure that they won't get AIDS. Thank you for watching, and I hope that you learned something about HIV reproduction and HIV anti-medications.